The human brain is complex. There's a lot we know about it, and there's still a lot left to learn. But there's only so much we can find out from traditional ways of studying the brain. So researchers at Novartis and other organizations are taking a different approach. They're growing brain organoids in the lab.
In the past, in order to study neurons, we had to use neurons from mice or from rats in order to have the cell type that might be affected in disease. However, mice and rats don't naturally come down with schizophrenia or Parkinson's, and so there's some limitations to what you can learn from studying rodent neurons.
And so we really need to be able to study the human brain if we want to understand the human brain.
But that's easier said than done.
It's been difficult in the past to acquire human brain tissue. So the brain is locked within the skull, and so it's very hard to get a biopsy, and then it's very hard to keep that tissue alive.
But this is all changing with this scientific breakthrough.
The breakthrough really came from our ability to use induced pluripotent stem cell technology to be able to grow an organoid in the lab. So an organoid is a three-dimensional organ that mimics a 3-month-old human embryonic brain. It has very similar structure where it has a ventricle, it has these cells which are called progenitor cells, which give rise to all of the neurons.
It's only a few millimeters in diameter, and it also cannot think or learn or process information. It doesn't have sensory input, and so it can't learn or form memories.
Even though these organoids are very young in development, they still have human neurons. They still start to form connections similar to the human brain.
The researchers build the organoids by taking skin or blood cells from a patient, reprogramming them into stem cells, and giving the stem cells cues to coax them to become neurons which self-organize and connect with each other. This provides a platform for studying brain diseases as they've never been studied before.
So the ability to grow human neurons in a dish now allows us to study the very cell type that is most affected in disease. We can study a neuron from a patient that has schizophrenia. We can study a neuron from a patient with Parkinson's disease to try to learn what has gone wrong and try to fix it.
The other very important aspect is that we can manipulate the system.
So we can use a technology called CRISPR gene editing in order to remove individual genes one at a time from the organoid. So we can take a gene that we think might be implicated in schizophrenia, remove it, and see how the organoid behaves.
And Novartis is breaking new ground by scaling up this induced pluripotent stem cell technology. Researchers at the company are using organoids to study brain diseases and discover potential treatments for them.
The work that's being carried out at Novartis is innovative because we are applying robots and automation to making organoids. We can now make hundreds or thousands of organoids.
Scalability allows us to screen for many, many different drugs. It's like looking for a needle in the haystack, where we need to screen millions or tens of millions of compounds. And so you couldn't do that manually.
We had one project where we wanted to generate neurons from 47 different people, some with schizophrenia and some without schizophrenia. And that would have been impossible to do without having the automation.
Brain organoids may ultimately unlock new therapies for diseases that have stymied researchers for decades.
This research approach is very innovative. It's right at the cutting edge. I mean, we are pushing the limits of what we can do at Novartis. It's a renaissance for biology. It's really a fun place to be, and it's a fun time to be involved in this field.